One of the important steps in the process of gene cloning, it is the purification of plasmid DNA. So you are well familiar now that if we want to transfer our gene of interest, then we need carrier molecules, and here we are discussing the plasmid. So it is a prerequisite condition to perform gene cloning. Isolation of plasmid, it is very tricky, you can say that step, because when we attempt to isolate plasmids, it may be contaminated with chromosomal DNA. Why we need this step? The reason is that even if we introduced our recombinant DNA plasmid, so sometime we want to analyze that whether our foreign DNA is inserted or not. So even then we can isolate plasmid DNA in addition to isolate natural plasmid. So as I told you that it is very tricky step and should be performed very carefully. So first step is that the lysis of the cell. So the lysis should be performed very carefully. The first step is that to open the cell wall that can allow the plasmid DNA to escape. Or lysis can be performed so that one can get the clear lysate. Lysate, it is a fluid that contain the content of the lysed cells that contain our plasmid DNA and the chromosomal DNA. And if this step it is performed carefully, then the plasmid DNA mainly remain in intact form, whereas the lysis can result in the fragmentation of the chromosomal DNA, because during the lysis step we can also apply shearing forces sometime that can cause the fragmentation of the chromosomal DNA. Now we get a clear lysate. How the plasmid DNA can be separated? There are different steps, but we can discuss two methods here. The first method, it is the isopicnic centrifugation. Isopicnic means that the compounds that have the same density, they can be called as isopicnic. And centrifugation, it is a process in which we can use centrifugal forces for the isolation of our compounds. To isolate the plasmid DNA from the lysate, we can centrifuge it in the presence of cesium chloride gradient that also contain ethidium bromide. What is the advantage of ethidium bromide? The advantage is that intact plasmid DNA can bind the ethidium bromide up to a certain extent, because the free DNA ends, they are not available. On the other hand, the fragmented DNA that provides the free end, it will bind more ethidium bromide so that its density will be lower, and the density of the plasmid DNA, it will be high. And when we will centrifuge the lysate in the presence of calcium chloride, rather cesium chloride gradient that contain ethidium bromide, then we can get the two bands of the nucleic acid. As indicated by this diagram, the two bands of nucleic acids, they are separated depending upon their density. As I told you that chromosomal DNA that is fragmented, it will bind more ethidium bromide that will lower its density, and it is present in the upper portion of the centrifuge tube. And on the other hand, the plasmid DNA that is intact in covalently closed circle form, it binds lower concentration of ethidium bromide because of its intact form. So it is having high density and is present at the lower side in the centrifuge tube. So this band, it can be purified by using different techniques like aerosol purification. So this is also one of the technique by which we can isolate the plasmid DNA from a clear lysate. Another method uses the narrow range of pH, where the chromosomal DNA, it can be denatured. That is, its double strands, they will separate from each other, and this range of pH, it is very narrow, and it is between 12 to 12.5. So at this alkaline pH, double stranded DNA, that is chromosomal, it will denature, and then we can neutralize the lysate. So because at high alkaline pH, the plasmid, they will remain intact, and when we will neutralize the pH by using acetic sodium acetate, then the chromosomal DNA, it will again renature, and it is converted into insoluble networks. Another cell debris like the proteins, RNA, they can also be co-precipitated by using sodium hydroxide or sodium dodecyl sulfate, or SDS. Now clear lysate contain the cell debris that contain the cell wall, different type of proteins, RNA, insoluble chromosomal DNA, and the plasmid DNA. So plasmid DNA, in the later step, it can be purified by centrifugation, agarose gel, or by using other techniques like the gel filtration. In addition to these two techniques, nowadays some commercial kits, they are available. By using that kits, plasmid DNA, it can be isolated within relatively short time. Now there are different factors that can affect the yield of plasmid DNA. The yield of plasmid DNA, finally, it may be higher or lower. Now which factors it determines the yield of plasmid? So one of the factor is that the copy number of the plasmid. If copy number it is high, the plasmid yield will also be high, and vice versa. In addition to that, what is the growth stage of the bacterial cells that you have harvested the cells? If you taken the cell from the log phase, where the cells, they are exponentially growing, you can get the high yield of the plasmid DNA. Whereas if you are taken other stages like the late stationary phase, the plasmid DNA, it may be present in lower concentration. And finally, in addition to the stage of the growing of the cell, of course it is also important, but the physical conditions that are used for the growth of microbes, they are also very important. Like if nutrient level it is low, the DNA yield may also be low, and if nutrients, they are optimum, cells, they are growing faster, they are entering into the log phase rapidly, so the yield of the plasmid DNA will also be high. So these are different methods by which the plasmid DNA it can be purified, and also the yield of plasmid DNA, it can also be influenced by different type of factors.